In our continued effort to move into a system level of organization, we have to talk about the tissues first. We discussed the cellular level, and if we can get the same type of cell adhered to each other in a mass, then we have a tissue. Then if we can get multiple different types of tissue together, then we potentially have the organ level of classification. Now, there are a couple tissues that we're not going to get into much at this point. You'll see here at the bottom of the screen, we have muscle, nervous, epithelial, and connective. The main reason we're not going to get into all of those four in depth at this point is because at some point we're going to get into muscle tissue as part of the muscular system. Almost all the physiology of the muscle system is going to be involved with muscle tissue. Same goes for nervous. All of our neurophys will revolve around discussions about the nervous tissue. So that leaves epithelial and connective without their own systems for us to get into in depth here as far as their classification goes. So you will need to understand the characteristic of each tissue type, and it's important to realize how the structure relates to function. In lab, you will get plenty of opportunity to see slides and pictures of tissue in which you will have to classify them based on what you see. Here, we're going to be more in touch with why does that structure lend itself to functions at those particular locations. So we've been talking about the tissues now, and as we get into the system level, we'll mention any number of organs. But when we talk about an organ, realize we're talking about at least two different types of tissue that makes them up. So to at least introduce the muscle tissue, we have three different types: skeletal, smooth, and cardiac. And as part of our eventual AMP1 discussion, we'll be considering skeletal muscle tissue as part of our skeletal system. Smooth muscle will come up when we discuss blood vessel and bronchial dilation with the cardiovascular and respiratory systems, respectively. And you will talk about cardiac tissue in depth once you do the heart. We classify each of these tissues and differentiate them based on two variables: whether or not that tissue is voluntary and whether or not it is striated or has a striped appearance. So you'll see that skeletal muscle is striated. It looks like a striped tissue. You'll see that on the slides. And it's also voluntary. You have voluntary control over its contractions and relaxations. Smooth muscle, part of the name gets itself from the fact that it is non-striated. It looks smooth as opposed to the rippled appearance that you see with striated tissue. But it's also involuntary. You have no active control over the contractions and relaxations of smooth tissue that will make blood vessels constrict and dilate. Cardiac will be striated. We'll at least share that physical feature with skeletal muscle, but it's also going to be involuntary. When you get to the heart, you'll actually talk about the heart being auto-regulating. You get an auto-rhythmicity to give us a nice basic heart rate of a normal rhythm. And how we influence that is through sympathetic and parasympathetic nerves to speed that base rate up or slow it down. Here's skeletal tissue. What we're looking at with skeletal muscle tissue is long, parallel bands of muscle cells. And if we use the word muscle cell versus muscle fiber, those are the same thing. But you can see the striped appearance. That's going to be the result of filaments deep in the muscle cell overlapping with each other. So where you're seeing the light bands, more light is passing through, implying there's less overlap. Where the darker bands are, we're getting more filament overlap. And when we finally talk about muscle tissue down the road, we'll talk about how those filaments slide over one another as the muscle contracts. In any given organ, we will typically see these long muscle fibers run from one end of the organ all the way to the other. In cardiac, we still have the striated appearance. It's a little bit more difficult to see, but there's a couple areas on the slide there where the striped appearance kind of shows through the tissue just a little bit more than the other areas. So we still maintain the striated nature. The overlapping filament structure deep in the muscle cell is very similar to skeletal. But unlike skeletal, we do not have long parallel fibers. We've got shorter fibers that are attached end to end. You can see the junctions here, called intercalated discs. As one cardiac muscle cell binds to its neighbor, so they're shorter in dimension. Now, not only are they shorter, but they branch. So you see one cell here with this intercalated disc, but by the time it reaches the other end of the cell, it has formed a junction with two other cells. That is not unusual, and it leads to a more effective tissue for the location in which it's found. In the heart, it's a muscular sac. This branching network of fibers allows, when the heart to contract, we get a three-dimensional compression on the chambers inside. Now, embedded in these intercalated discs, we've got another adhesion called a gap junction. Simple protein tubes. And what that will allow for is that when we get a contraction of a muscle cell here, exchange of intracellular material through those gap junctions will give us the next muscle contracting. Therefore, it'll give us the next muscle contracting. That's going to be one of the main differences functionally from skeletal muscle. A skeletal muscle cell, every single one of them in an organ, requires a neuron telling it what to do. In cardiac muscle tissue, we can have an electrical impulse at one site, and then we will see a wave of contraction spread from that one location across the tissue, leading the entire heart, per se, to contract based on that single stimulus. Smooth muscle, as you can see, we see no areas that look striped or striated whatsoever. Hence the name smooth. Now, we will typically see smooth muscle arranged itself in a circular manner around orifices like we have on the slide here. So picture this could be a blood vessel, and as they constrict, this blood vessel will narrow down. There could be a bronchiole in the lungs. This could be the opening to a duct that's opening and closing. This will be the type that we talk about structurally the least. Now, as far as functions, obviously that's going to depend greatly on where we find the tissue, the type. The functions of cardiac muscle tissue versus smooth versus skeletal are going to vary dramatically, and it's going to be very specific for the system in which it's found. For nervous tissue, we kind of step outside the box of our tissue definition just a little bit, because we don't have a single type of cell adhered together in a mass. We've got a number of different types of cells working together as a tissue. The primary cell is going to be the neuron. If you picture what the nervous system does, you're picturing what the neuron does. Sending electrical impulses to one part of the body to another, like the wiring in the walls. The other cells that are associated are all supportive to the neuron. They all help the neuron do its job. And as a whole, collectively, we call them the glial cells, implying that they're supportive. And they all play a role in maintaining the neuron. The astrocytes maintain the chemical environment around the neuron. You'll see once we talk about neurons, it's all about the chemistry and the movement of items across the membrane. They're very sensitive to changes in their chemical environment. Astrocytes are going to be responsible for maintaining that environment and cleaning it up. Microglia. we're going to be referring to the microglia as the immune system cell of the nervous system. We have Schwann cells and we have oligodendrocytes. Now, functionally, we're not going to differentiate between the two. They both accomplish the same function. They help neurons transmit their impulses faster. The difference is we will see Schwann cell in peripheral nerves, nerves that run down the arms, the legs, the nerves that run into the abdomen. Oligodendrocytes are only going to be found in the spinal cord and the brain. So same function, speeding up transmission, location is going to be different. And then finally, ependymal cells. Ependymal cells are cells that line cavities inside the brain, and they produce cerebrospinal fluid. The nervous system is going to be one of those excitable tissues, and we will be referring to it often as we go through the next several months of classes. It's an integral part of our feedback loops, which of course homeostasis is going to be an ongoing theme. We rely on the nervous system as part of our communication network. It helps us monitor changes in the body, and it helps us recruit organs to respond to those changes. So enough of the tissues that we'll get into later. Let's get into the ones where we'll be handling them in depth now. Epithelial tissue. This is a covering and lining tissue. Anywhere where the internal body meets the external environment, it will have a layer of epithelial tissue. Skin is an epithelial tissue, or parts of skin is epithelial tissue. The lining that goes down inside the mouth, the esophagus, lining the stomach, small intestine, large intestine, epithelial tissue. Down the inside of the trachea and the lungs, epithelial tissue. All of these are areas where the internal body meets the external environment, so it will have a lining of epithelial tissue covering it. Nothing gets into the body without passing through an epithelial membrane. But we'll also see epithelial tissue lining cavities. We mentioned membranous cavities earlier on in chapter 1. There are synovial cavities with several of your joints. All of those will also be lining the inside of those cavities. Epithelial tissue takes on the function of absorption and secretion. Obviously, we just said if anything is going to pass into the body or out, it has to go through an epithelial lining. So if we eat something and we want to absorb it, it has to go through an epithelial lining. Gases that are exchanged at the lungs, an epithelial lining. It's also going to form a frontline protection. We just talked about a couple things that we want to get in and out of the body. What about things we don't want? Bacteria, viruses. We'll also use them as a form of secretion with our exocrine glands. Keep in mind, we don't mean endocrine glands. Exocrine glands are going to be associated with the integument system, like we said. You'll see it in that chapter. Producing oil in the hair follicles, sweat that gets secreted onto the surface of the skin, mammary glands producing milk. Makes a product, secretes it onto the surface of the body. Endocrine glands produce a very particular product called a hormone that gets secreted into the blood. Now, for epithelial tissue, we see several different types manifest based on two different variables. So whenever you're given a picture or a slide on a microscope from a tissue, you need to be able to look at these two variables. The first one is how many layers. Now, luckily, we're not going to be asking you to count the layers, because some of them are going to have hundreds. So all you need to differentiate is there one layer or is there more than one layer? Those are your two options. If there's only one layer, we give that particular tissue you're looking at a descriptor of simple. If there's more than one layer, we call it stratified. The other variable is the shape of the cell. We have three options: squamous, cuboidal, and columnar. Now, two of those, hopefully the name speaks for itself. Columnar, coming from the word column. They're taller than they are wide. Cuboidal, shaped like a cube. They're about as wide as they are tall. Now, please don't look for nice geometric features here. These are still going to have somewhat rounded edges. We're comparing these shapes to the overall dimensions of the cell, not nice 90-degree corners. A squamous cell is a broad, flat cell, extremely thin. So we look for the number of layers, then we look for the shape. Now, as far as shape goes, there's only one place we look: the superficial layer. If they're simple, now obviously we only have one layer. That's the shape of the cell we have to deal with. But if there's multiple layers, we only look at the shape on the most superficial layers. And we use these two variables to mix and match to give us our tissue types. So we can have simple squamous, stratified squamous. Simple cuboidal, stratified cuboidal. Remember, epithelial tissue, we mentioned this in integument as well, is always avascular. There is no blood supply going through epithelial tissue. So let's look at how this will present. Here is an example of simple squamous. On the left, we have a drawing of what is showing on the slide on the right. Single layer of squamous cells. They're broad, they're flat. We've determined they're squamous. Right? A cuboidal cell will be more along these dimensions. A columnar cell will be taller than it is wide, right? Don't make this mistake that this is some sort of column laying on its side. We consider when it's wider than it is tall, that's broad and flat. Now, to get us in the right mindset of how we want to tackle this material for lecture, we're all about why the structure makes it great for function. And what we have here is a single layer of broad, thin, flat cells. So ask yourself, what will this tissue be good at? Is this the type of tissue you would like to depend on to resist a lot of physical wear and tear? Hopefully you said no. It's a single layer, and the cells that you have are extremely thin and fragile. You're going to want this, if it's going to be anywhere, to be in a place that doesn't get a whole lot of physical abuse. But what that thin dimension of the membrane gives you is very easy transport. Remember, we talked about diffusion in one of the last chapters. And one of the things that determined the rate of diffusion was the distance something had to travel. Well, if we have to be passing things through this tissue, you won't get a thinner membrane than that of simple squamous epithelial tissue. So, thin, flat doesn't give us a whole lot in the way of physical resilience, but it's great for transport. Doesn't that sound like something that would be ideal for gas exchange at the lungs? That's where we want to be looking here. Here's what this would look like if we were looking straight down on it, almost like a flagstone type of walkway, if you will. Here we have our next type. Now, hopefully by looking at that, you see that we have one layer. You can distinctly see how this row of cells is distinctly different looking from the underlying tissue. One layer. What we have here is a cuboidal cell. Now, when you're looking for these in the lab, you will typically see them arranged in circles. It's not unusual to see these cuboidal cells form tubules, like we see in ducts, particularly at the kidney. Here's what a columnar cell will look like. Now, I said earlier, it's not going to look like a nice geometric shape, right? But that cell is distinctly taller than it is wide. That's a columnar cell. One distinct layer, as we can see a different type of tissue underneath it. In the digestive system, we have a lot of mixed function. We're going to have some secretion, especially in the stomach, secreting acidic contents. We have to have a cell that's great at absorption, but also it's going to get some physical resilience. The absorption is aided by the fact that we only have one layer, because even though the cell is thicker, we got a plasma membrane to go through here, we got a plasma membrane to go through there. So it still only has to get through one cell layer. But the dimensions of the columnar tissue is going to give us more resilience than a simple squamous. Now, we don't see stratified columnar very often. What we tend to see is a pseudo-stratified. So you can get rid of stratified columnar. Even though this tissue looks stratified because of the staggering of the nuclei. But on the drawing, it's a little easier to see. You see cells. Now, some of them don't make it all the way to the surface of the tissue, like that one. But every single cell does attach or touch the basement membrane. Every cell touching the membrane means we don't have any cells stacked on top of their ones underneath. This makes it a single layer of cells with the appearance of stratification. Therefore, pseudo, meaning fake. Now, where we do see stratified tissue very readily, probably our best example of stratified tissue, is with squamous. You will see a great example of this when you look at the epidermis in the skin. Here's where the tissue starts. If I can get my marker working. So this is one of those examples where thankfully we don't have to count the layers, right? But obviously there's more than one. That makes it stratified. But it's also a great example of why we need to make sure we're looking at the superficial cells. Remember, this is avascular, no blood supply here. The blood supply to this tissue is underneath. So the further these cells get from the blood supply, the more their shape is going to change dramatically. So down deep here, you will have cells that look maybe spherical or cuboidal, but by the time they reach the surface, they're broad, flat, and thin, and actually not even really living cells anymore. Always look at the surface. Stratified columnar, stratified cuboidal. We don't really see that one very often. But again, we're looking for layers, then we look for the superficial shape. That will give you your combination of classification. Transitional epithelial is a special case. Normally in class, I would circle this with my laser pointer and have you guys take a guess at what type. And several of you will look and say, oh, there's more than one layer, so it's stratified. And if we look at these cells here, if I had to pick 1 to 3, cuboidal. So you'd call it stratified cuboidal. Then I would have you do the same thing for this tissue. And you would look at the number of layers and consider it stratified. Then you would look at the superficial cells and call them squamous, stratified squamous. Now, just based on what you can observe, I would not argue with that at all. However, now I tell you that this tissue at the top is the exact same tissue as the tissue on the bottom, except we're looking at them at different times. Transitional epithelial tissue undergoes a lot of stretch and retraction. They'll be found lining organs where that's part of their natural function. And because of that, what the tissue undergoes physically keeps us from being able to use our normal classification system. We want a classification system that's absolute, that isn't dependent on when in the day we look at it. So think about organs that undergo a lot of distension and retraction. Urinary bladder and the uterus. Some people will say stomach, but the stomach does not undergo as much distension and retraction as those other two organs. And most of their changes in volume will happen over long periods of time, not this short extension and retraction as part of its distinct function. So just for some practice, you can look at these slides. What tissue type do we have? One layer of columnar cells, simple columnar. Multiple layers of squamous cells, stratified squamous. Now, here we see some arrows pointing at some accessory structures. On columnar cells, we can see two manifestations of accessory structures. Cilia, which are actually what we see here, and microvilli. Cilia are hair-like projections. We'll find them on the columnar cells of the upper respiratory tract. They stick out into the mucosal lining. You inhale some particulates, it gets lodged in the mucus, and you can use the cilia to work it back up out of the respiratory system. Microvilli are finger-like projections, and we'll see those associated with the intestines, the intestinal wall. Those finger-like projections will extend out from the surface of the columnar cells there, increasing the surface area of those cells, increases the availability of the surface area for absorption of whatever you're trying to digest. Now we go through the connective tissue, and we're looking at this in the exact same way. We want to make sure structure versus function. Now with connective tissue, obviously, it's going to be structured a lot differently than what we saw with epithelial. These are supportive and protective tissues. So as part of an organ, you will see connective tissue act like the glue that binds it all together. And out of all the tissues we'll talk about, connective tissues will be the most varied. They can be soft, liquid, pliable, hard, rigid. Blood is a connective tissue. Bone is a connective tissue. So you see the full spectrum of physical appearances. In between there, we have things like adipose tissues, which is soft and pliable, varying pliabilities that we see in the different types of cartilage. Generally speaking, here's the classifications of connective tissue we're going to discuss: loose, dense, and then adipose. When we think about connective tissue, those different types, blood, cartilage, bone, everything in between, there is a consistent structure. Each of those varying physical types have a cellular component. Now, the cells in blood are not the same as the cells in cartilage, which again are not the same as the cells in bone, but all of those different types have a cellular component. They all have a fibrous component. And again, the fibers seen in blood for blood clotting are not the same as the fibers seen in cartilage, and they're not the same as the fibers seen in bone, but they all have a fibrous component. Then suspending the two, we see this ground substance or matrix. So an analogy that I've been using for years is this old-fashioned Jello molds. Make them like a bundt pan. Back in the day, they put bits of fruit in the Jello to make it look fancy, and then when you tipped it out, you saw the fruit particles suspended in this Jello. Well, now picture you're going to make one of those, but you're also going to put spaghetti noodles in there. The fruit bits are going to represent the cells. The noodles are going to represent the fibers. And the jello itself represents the matrix that suspends the first two. The matrix is going to be the component of the three that directly gives us this distinct difference in physical appearance. Liquid, bone, anywhere in between. Because if we take that jello mold and we leave it at room temperature, it jiggles. It's a semi-solid. Maybe like adipose tissue. A little bit firmer, like cartilage. If we take that jello mold and put it in the freezer, it will get rock hard like bone. All that really changed was the consistency of the matrix, the jello. If we take that same jello now and put it in the oven and warm it up gently, it will melt back to a liquid. Again, the fruit and the noodles are still as they were. What we really changed was the jello, the matrix. So the matrix itself gives us the varied physical appearance. Based on the drawing here, here's some cellular components, here's some fibrous components running through, and of course all that beige background is going to be represented by the ground substance or matrix. Here's an example of a loose connective tissue. Now, when we use the word loose versus dense, we're referring to the density of the fibers. Here you can see a lot of fibers, or a few fibers, suspended in a lot of ground substance matrix. The fiber to matrix ratio, so to speak, is low. Now, this is going to give us a tissue that's very pliable. Maybe not that strong, but very pliable. Here in the little key on the right, you see that it's going to make up a lot of the dermal layer of our skin, particularly that upper portion of the papillary dermis. Think about every time you bend your arm, the skin over the point of the elbow has to stretch. The skin on the inside of the elbow has to compress, and you straighten the arm out, and we expect it to ply right back to the way it was. Areolar tissue gives us that ability. Here we have some adipose. Now, adipose is going to be an extremely loose tissue. Very little fibrous component, period. Cells basically filled with fatty substances as a storage vesicle. And here we have two examples of dense. Dense regular, dense irregular. Dense because we have a lot of fibers. In each of these examples, basically all we see are fibers and cells. Very little ground substance. Regular implies the orientation of the fibers. We have this parallel arrangement in regular, whereas in irregular, they're kind of three-dimensionally woven through that tissue. Now think about what that will give you structurally. With all of our fibers oriented in the same direction for regular, gives us maximum resistance in one direction. Think about the Achilles tendon at the back of your ankle. Safe to put all your eggs in one basket there, so that any resilience in this plane, all of these fibers are resisting that particular stress and strain. Dense irregular tissue would be ideal for any tissue where we got a vast assortment of stresses and strains coming at us from all three dimensions. Dense irregular will never be as strong as dense regular in any one direction, but it is going to be more diverse about resisting stresses and strains in multiple directions than dense regular will. So know the tissue type, how to classify, and then know how the arrangement meets the structural demands. So we'll be asking you questions along the lines of, Which of the following tissues is best suited for fill in the blank? Pick your choice, pick your option and fill in the blank. Which of the following tissues is best suited in a area that undergoes very little physical abrasion but demands a lot of ability to absorb? Simple squamous. Which of the following tissues is best suited against extreme stress and strain in any one direction? Dense, regular connective tissue. So that's how we're looking to tackle this topic in lecture as opposed to lab. Structure versus function. In lab, you'll be much more likely to get a slide, and it'll simply ask you, what is it? Where do you find it? What does it do? Those will be the 3 questions you'll have to know in lab about any particular tissue type. So let some of that digest. On exams, you may get 1 question on muscle, you may get 1 question on neuro, but the bulk of the questions in this particular section will come from connective and epithelial tissue. Understanding at this point, any mention of muscular and neuro was only done to round out this topic, because you'll be sick of listening to muscle and neuro tissue once we get to those relevant systems.